Good morning. We are going to get started. We hear that the traffic is pretty bad this morning, so there will be many people joining us once we have started, but it's important not to delay everything that you are going to be hopefully enjoying after our lectures today. And without any further ado, it is my pleasure to welcome Professor Paul Laisel, our principal, to welcome you for today.
Thank you, and well, good morning, everybody. Welcome to Royal Holloway. I'm going to put that there. So, the history of this university goes back to 1886, when Thomas Holloway and his wife Jane opened a university that was open to women to come and study. And at the time, largely women couldn't go into university. It seems unbelievable in this day and age. And they laid down some of the foundations that ensured that today women and men are treated equally, at least in terms of coming to university. Their initiative was something new and bold and pioneering. And that tradition lives on today when we have students that come from all over the UK and 120 countries around the world. And we do a lot to help our students, as they did back in 1886, to make the transition from college and school into university life, giving particular help to those students who have additional needs, or for those who are going to university for the first time from their family. Today we continue that pioneering spirit of Thomas and Jane by not only teaching our students the very latest developments in the subject, but as one of a handful of universities, we also do research that helps to create those new ideas and developments. So our teaching staff are working on scientific breakthroughs and finding new ways to think about their subject that makes them world-leading experts in their field. And today you'll be hearing from some of them and some of the work that they're doing. So the theme, of course, is rare disease and Rare Disease Day. It affects some 7% of people at some point in their life. That's around 3.5 million people in the UK. And today is part of celebrating Rare Disease Day, an international day that raises awareness of the many diseases that we don't necessarily hear so much about in the media and press and online. I think traditionally it's supposed to be the 29th of February. That's a day that doesn't happen very often, so when there isn't a 29th of February, it's towards the end of the month. This is the 12th occasion in which this event has been held. So I hope today that you will be informed and inspired by the work that goes on here and in other places on tackling rare disease. And you'll hear particularly about gene and cell therapies, treatments for some rare diseases that are being developed here at Royal Holloway. And maybe one day you'll be here giving the lecture. I hope it inspires you and makes you want to work in this very important area. So the theme is bridging health and social care, and I'm going to pass over to Raphael, who's going to say a little bit more about the programme for today. So once again, welcome to Royal Holloway and enjoy the day. Thank you very much.
So good morning, everyone. Can you hear me well? Okay, that's great. As Paul Laisel has kindly introduced, we are very keen to celebrate the Rare Disease Day here at Royal Holloway for a variety of reasons, which I will introduce. I will also give you an idea of what you are going to be doing during the day, and mainly really why this is so important. I think it's important also to for you to be clear of what are my credentials to be discussing this topic with you today. So I have a variety of roles. My two professional ones, so to speak. I'm a professor here at Royal Holloway, and I'm the editor of a journal called Gene Therapy, which is one of the leading journals in this particular scientific field. But I also have two honorary roles. I'm the treasurer of one of the learned societies in the field, the British Society for Gene and Cell Therapy, and I'm the chair of the Genetic Alliance UK. It will be my pleasure today to introduce the next speaker in a few minutes, who is actually the chief executive of this charity, the Genetic Alliance, and she will tell you how important this umbrella organization for patient societies is. This is an outline of my lecture today, so you know what I will be discussing. What is a rare disease? What is this day? Why is this important at this place, Royal Holloway? I will take you back a little bit about genes and diseases to remind you the molecular basis of this, and we'll discuss a little bit about mutations, and then the progress that is being made, but also some of the very important, perhaps social issues in the field, the pricing of the medicines that are coming along. And I'll finish with a note on the inspiration that we can draw from all this. So a little bit as Paul was introducing, there is a way to define rare diseases, and different countries or different groupings of countries have a different definition. But in the European Union, a disease is rare if fewer than 1 in 2,000 people are affected. The issue is that there are many rare diseases. We don't actually know how many. Many are actually being described on a weekly basis. So there are somewhere between 6,000 and 8,000. They affect 7% of people because there are hardly any treatments for these people. They take a disproportionate part of the health budget, about 20%. In addition to that, most of those diseases affect children, and 30% of those will die before age 5, which is terribly sad. And most of us really do not know about this. So something really important is to raise awareness about all this. There is a list of rare diseases, as you can imagine, updated very, very regularly, and these are the first 10 diseases on the list. What I want to give here is just a sense of how many there are. So is anyone brave enough to try to guess how long will it take to read the rare disease list aloud if you were to attempt to do that? Literally just reading one after the other. How long would that take? 2 hours? Okay. Any more? Go on. Any more guesses? Perhaps a little more? 3? Okay. I'm going to spare you the suffering. It would actually take 12 hours just to read the list of rare diseases if you were to do that. And if you imagine that people can actually devote their whole life to trying to understand and develop a treatment for a rare diseases, that can give you an idea of the task that is ahead of us. In addition to that, these diseases are important because in many cases we know the therapeutic target. We know what we would have to do in order to try to cure that disease, which is not true for many common diseases. And furthermore, a lot of the technology that is being developed in the context of rare disease to treat these diseases is being applied to more common diseases, highlighting further their importance. So what is Rare Disease Day? Is a day that takes place on the last day of February each year. The reason being that on leap years you have the 29th of February, which is a rare day. So on that, people decided to hold Rare Disease Day on the last day of February every year. This year is the 12th edition, and the main objective, as I said, is to raise awareness amongst the public, decision makers, and many other stakeholders, as you can see on that slide. And why are we here at Royal Holloway so keen to celebrate the day? Well, the greatest part of these diseases are inherited, and for them, maybe using genetic and stem cell technology, you can actually treat them. At Holloway, we have an international reputation in this field, so we are very keen to showcase our research and our teaching in this type of outreach activity of which you are taking part today. There are a group of us working in this particular area, and particularly Professor George Dixon will be speaking, will be doing the last lecture today. So you will have an opportunity to hear some of the actual research that goes on here at Holloway in this field. We have very recently set up a center of gene and cell therapy, and our mission statement for this center is to deliver research, teaching, and outreach, like today, in gene and cell therapy, with a focus on rare diseases of the neuromuscular and central nervous systems. I told you that there are loads of rare diseases. We cannot work on them all, so we concentrate on some of them, like muscular dystrophies, spinal muscular atrophy, ataxia telangiectasia, motor neuron disease, but as I mentioned, the technology is also applicable to more common diseases, so we are also working towards treatments for spinal injuries, Parkinson's, Alzheimer's, even antibacterials and antivirals. So taking you back a little bit, I keep telling you that many of these diseases are inherited. So this is a karyotype of the human genome with a particular gene highlighted in green, which you can just about see. And there may be about 25,000 of those on a very rough count. Each of those genes will normally encode a protein, will have the information to produce a particular protein. If all is well, you have a normal gene and you have a normal protein, but perhaps the gene has a mutation, represented here by that green blob, and then that gene will produce an abnormal protein or will produce no protein. And what can be the outcome of that? Some mutations are irrelevant. They really have no effect, or maybe a particular gene covers for the mutant one and there is no obvious phenotypic effect. Others are minor, so yes, there is an effect, but it's relatively possible to cope with that. And evolution is based on mutations, so our capacity to adapt to a changing environment is based on mutations, so they are important. But in some cases, a mutation can have a devastating effect. And as examples here, I'm showing images of people affected by Duchenne muscular dystrophy and spinal muscular atrophy, both of which are lethal diseases. So mutations in the genes responsible for these diseases can lead to an early death. We have mentioned that it's very important to raise awareness. One of the issues with these rare diseases is that diagnosis can take a long time. In social media lately, I heard that a particular person took 18 years to be diagnosed of the rare disease accurately. So it's a lifetime, and that obviously is a very important issue. But also often these people need very many different types of care, and coordinating that care, which is something that may seem obvious to everyone, is actually not current practice. And to achieve that coordination of care is one of the goals of the day this year, and is part of the theme. The Genetic Alliance UK and their campaign Resist Rare Disease UK is very active in this area, trying to highlight this need. So is there hope in this massive field of importance? Well, yeah, there are more and more successes in gene therapy clinical trials where new therapeutics are tested in people, and a handful of them have been marketed, and I'll show you a few of those in a minute, in a number of diseases. So both clinical trials and marketed products in these areas, successes in clinical trials in this, and as I was telling you, even application to more common diseases like leukemia for some of the technology that has been developed in this area. And there is an international rare disease research consortium which, given the success in the initial goals that were set up, updated their vision for 2017 to 2027 in such a way that they aim to diagnose all rare diseases and develop 1,000 new therapies by 2027. So there is a lot of work going on and significant success being achieved. what does success in this may mean? Well, this is a real child, a drawing, but it's a real child. His name is Cameron. He's affected by spinal muscular atrophy. And all the children affected by the most severe type of spinal muscular atrophy, type 1, would die before age 2 if they are not treated. And recently, a marketed drug became available, and Cameron and many other children were able to achieve developmental milestones like sitting up unaided and standing up that were unthinkable before. So these new treatments can have a massive effect on people affected. They may not be entirely a cure, but they may have a massive therapeutic effect. These treatments currently come in 3 flavors. So we can engineer viruses in order to deliver genes that can have a therapeutic effect, or we can genetically modify cells so they have a new gene that can have a therapeutic effect, or like in the case of the drug for spinal muscular atrophy, they can be a short nucleic acid that again can have a therapeutic effect. But if you can see the prices, even though they are in euro, there is more or less parity. So where it says euro, you can put down pound. They're massive. So how society is going to cope with the pricing of these treatments that are being developed, when we hear every day that the NHS is at crisis point, is a very important societal issue. So you are here listening to our lectures today. After the lectures, you are going to be attending an exhibition, and you will have a hands-on lab activity to learn a little bit about the inheritance of genetic diseases. And you will have a speed dating activity with undergrads to discuss in small groups a variety of topics of relevance. And you will have a disease detective activity in which you will try to guess what it is that is affecting a variety of people. So a variety of activities during the day which we hope you will enjoy. Just to wrap up, I want to highlight how inspirational the achievements of people affected by rare diseases can be. Over the years, we have had many people affected supporting us. This is Helene Rainsford, who was our patron in 2012, a Paralympic gold medalist. And this is Srin Madipali, who has spinal muscular atrophy, and he was the co-founder of the disabled Airbnb and recently moved to California to develop that company, a person with 2 degrees, really unstoppable. For an event like this to happen, many people have to collaborate, so it's only fair to thank a variety of university departments who help us to run the day, as well as the people, both the staff, postgraduate and undergraduate students here at the university, our exhibitors and our speakers. And you're here to learn. You're also maybe making your university choices. This field is incredibly active and exciting. We can now make defined changes to the human genome, hopefully with a therapeutic effect, and medicine is changing. Genomics is entering the NHS, will be in clinical practice very soon, and in artificial intelligence and the handling of the data that are being generated is going to be part of it. So how we understand medicine is changing massively. It's a really exciting time, and I hope that some of you will be enticed to, in some way, participate in this biomedical revolution. So thank you very much. And it is my pleasure now to introduce the second speaker for today, who is Dr. Jane Spink of the Genetic Alliance UK. Jane.
We started at the end.
Oops.
Great.
There you go.
Thank you. Good morning. It's great to be here. Thank you for the invitation. I'm Chief Executive of Genetic Alliance UK, and I'm also Chair of Rare Disease UK, but the thing that doesn't appear on my CV is that I'm the parent of a young man diagnosed with a rare condition at 14 weeks of age. and I don't know how to operate a PC. So Genetic Alliance UK is a national charity. We're a coalition of over 220 voluntary organisations, ranging in size from the very small right up to the British Heart Foundation, who are our largest member, and we exist to empower patients and patient organisations to transform the daily lives and future prospects of people affected by rare genetic and undiagnosed condition. You can find out more about our work in the exhibition later, and I've already mentioned Genetic Alliance UK is the parent organisation, and we have two projects, one already mentioned by Raphael, which is the Rare Disease UK campaign. Rare Disease UK has its 10th birthday this year, and it was set up to make sure that the UK government met its obligation to have in place a plan to tackle the challenges of rare diseases by 2013. And after that successful campaign, we've continued to campaign on behalf of our community to improve, for example, access to diagnosis, care and treatment, as well as to social care. Rare Disease UK has over now 2,500 individual supporters. It has membership of around 300 patient organisations and the support now of 27 industry partners. Finally, we have a project called SWAN UK. Now, SWAN stands for Syndrome Without a Name. Every year, around 6,000 babies are born with a complex condition of likely genetic origin. Around a third, as Raphael has said, will not make their 5th birthday, and around half, using current technologies, will remain undiagnosed throughout their lifetime. We provide 24/7 peer support for around 2,000 families across the UK. So what's the difference between genetic rare and undiagnosed? Well, it turns out very little. A rare condition has a prevalence of less than 5 in 10,000 individuals, so that's 1 in 17 of us. If you know more than 17 people, this should matter to you, whether or not you personally have a diagnosis of a rare disease. A single rare disease might only affect 1 or 2 individuals across the UK, but may affect up to around 30,000 people in the UK, and some are so vanishingly rare that you certainly won't have heard of them, because there are around 7,000 rare conditions, and we've counted that around 5 new conditions are being identified each week. And 80% of these turn out to be genetic in origin. Now, how do people find out if they have a genetic condition? Well, one way is through screening. And it is possible to find out if a baby has a genetic condition through what's called the blood spot screening. And when you were tiny babies, you will all have had a small amount of blood taken and placed on a card that was sent off for testing. Now, that was introduced in 1969 with testing for PKU. And for the next 30 years, no other conditions were added to that list. Since then, there have been only 8 additional conditions added to the list, and this compares extremely unfavourably to other high-income countries. Across Europe, newborn screening typically screens for around 20 to 40 inborn errors of metabolism and other genetic conditions, and in the States, up to 50 conditions can be screened for. We are currently undertaking a piece of work called a patient charter, which has looked at the methods that we currently use and what might be possible, not only through conventional approaches, but also through whole genome sequencing. And we know that there are around 1,000 actionable findings if you sequence a newborn's DNA that, if you act upon them, can lead to improvements in health and well-being. The other way to find out that you have a genetic condition is through diagnosis. So that's after you have developed symptoms. And there are huge challenges here because with the large number of rare diseases, it is simply impossible for a single GP or even a specialist to know all about all of those diseases. So if you go along to your GP, the most likely things are the things that will be looked at first. So you might have symptoms that are similar to a common disease, or you might have symptoms that seem confusing because they're not typically associated with a particular rare disease. And all of this compounds to the point where we found that on average rare disease patients consult with 5 doctors, wait 4 years, and in that period receive 3 misdiagnoses. As you can imagine, living with this so-called diagnostic odyssey can be very stressful, and there can be some real problems for individuals around practicalities. So, for example, accessing educational support, accessing care and treatment, or even simply being believed. It's not unusual for parents to be dismissed as neurotic parents when no definitive diagnosis can be found. The longest diagnostic odyssey that I've personally heard of was a man in his 50s who had experienced ill health throughout his life. Upon diagnosis, he said his life was transformed, and one of the reasons for that was that he was able to find out information about his condition, he was able better to maintain his health, and importantly, he was able to find a community who were affected by that condition also, and through that, he felt less isolated and more able to cope. Once you have your diagnosis, the challenges don't end there. Many rare diseases, many genetic diseases affect a number of different systems in your body, so you might not only have to see a cardiologist, you might need to see a neurologist, you might need to see a psychiatrist, you might need to see a dermatologist, and very rarely are all of those appointments coordinated, which means that on average rare disease patients might have to travel up to 2 hours to an appointment and attend at least 3 separate clinics, not always in the same hospital, and that happens at least 4 times a year. In terms of research, we know that rare disease patients are incredibly supportive of research, and indeed many of the charities who are our members support, promote and indeed fund research. So thinking of gene therapy, some of the initial studies that happened, particularly in the UK and the US, were funded and promoted by rare disease charities. We have 7,000 rare diseases, but only 5% have an effective treatment or cure. So that's 95% of rare diseases have no targeted therapy. So there's a huge unmet need. Patients and patient groups can help researchers in other ways. They can engage with researchers and companies in terms of trial design to make sure that when a patient volunteers to be part of a clinical study, that there are no elements of that study which are going to impact their willingness to continue. A big problem for clinical trials is dropout. If you don't make your numbers, you don't get your data. So what you want to do is enable patients to remain within that study and to contribute their data. They can also provide lived experience that can support researchers in designing treatments that meet needs, but also in terms of evaluating potential care and treatment once we have, for example, a licensed product. Now, there are around 140 products which have been licensed by the regulator, which is the European Medicines Agency, which means that they can be marketed and used in patients and sold to the NHS and other bodies. Half of licensed treatments are not routinely funded in the NHS across England, Scotland, Wales and Northern Ireland. So what this means is that once a drug has been proven or a medicine has been proven safe and effective, there is a hiatus of a number of years whilst it's being made available elsewhere that patients in the UK don't get access. What that means to patients in the UK is that they're having a suboptimal treatment, and during the course of those months and years their health deteriorates, and there have been clear cases where patients, including children, have died for want of access to a treatment that could have saved them. Our patients and our community, here are some quotes, and you can see that people feel let down and frustrated. And I think it's very difficult to understand why this should be the case, why in a high-income company there should be restricted access to effective treatments for patients with rare diseases. And yes, some of them are astoundingly expensive, but that's not the case for all. And we're working to put forward proposals that look not only at the cost of medicines, but also in the way that we fund and evaluate that medicines, with a view to improving that access to rare disease treatments. One of the problems is the complexity of the way that we assess medicines. Across the UK, we've counted 17 different decision-making points for a rare disease medicine, which is quite ridiculous for a drug that might affect only a handful of individuals. And also, an approval in England does not mean that if you live in Scotland, you'll be able to access that medicine, and vice versa. So what this leads to is active campaigns by patient groups. Here are 4 protests being carried out by our membership and in relation to treatments for mucopolysaccharide disease, for DMD, for cystic fibrosis, and for tuberous sclerosis complex. And these are all instances where decisions on access were delayed or extended. So it's Rare Disease Day, well, almost, and in the UK, Rare Disease UK is the official partner of Eurodis, who are the international lead for Rare Disease Day. So what are we hoping to achieve as a community? Well, we would like, through raising awareness and through spreading information about the problems associated with having a rare disease and accessing diagnosis, for example, and care, to create an awareness that something needs to change. So we'd like to see early diagnosis as the norm and not the exception. We would like everyone with a rare disease to have a personal care plan that is coordinated and to have access to all the information that they need to manage their condition. We want patients, their families and carers to be listened to and consulted, and we want research to deliver treatments that transform lives. And once that research has delivered a new treatment, we want every patient who would benefit to have access to the treatment, care and support that they need. So we've already launched our Rare Disease Day social media campaign, and I'd like to share our promotional video with you. Okay, now this is a challenge. It's so good you should watch it again. Let's see. I'm a Mac user, so this is like driving a left-hand drive car. Thank you. So here are some of the stars that we already have on our wall of stars. We have around 500 contributions. If you would like to contribute, if rare diseases affect you or someone you know, and you want to share your message through our campaign, please visit the Rare Disease UK website, where you'll find information about how to submit your star and how to share it through social media. And if you have a trawl through there, you'll find my own star. So thank you very much, and I do hope you will visit our exhibition stand.
And finally, it's my pleasure to introduce Professor George Dixon, who is going to show some of the research that is going on today as we speak at Royal Holloway. George, thank you.
Thank you. Thank you, Raphael. Good morning, everyone. Can you hear me? Okay, you understand this strange Scottish accent? Yeah. So I wonder, does anybody know, we've seen this picture already, did anybody tell me what is the tissue that I'm showing here in this slide? This striated tissue, any ideas? The clue is in the title, actually. Yeah. Muscle, what type of muscle? Skeletal muscle, great. So yeah, skeletal muscle. We're talking about disease today, but most scientists who are working in this area, they come to it from appreciation of the wonder of nature. I work on skeletal muscle. It's a wonderful tissue. You can see 4 skeletal muscle cells here in sort of longitudinal visualization, and the blue dots are nuclei. And your muscle cells, some of them are maybe up to 10 centimeters in length, single cell membrane with perhaps 10,000 nuclei inside each cell. Isn't that amazing? Not what you sort of see routinely in the textbook. So even you have some wonders of nature there. So skeletal muscle and the wonders of science. I'm here to talk today about delivering a gene therapy for a set of rare diseases called the muscular dystrophies, and our ability to do this is based upon, as we've heard from Jane, from Raphael, this amazing technology of sequencing DNA and the computing power to analyze that stored sequence on a massive scale, which allows us to understand the molecular basis of these diseases, allows us to search then for new treatments, and also allows for very precise diagnostics and prediction risk. And actually, if you're having a debate in school at all in the near future, I would suggest you have a debate on something called preconception diagnosis. I won't describe it to you, just think it through. Preconception diagnosis. Parents being genetically analysed for the risk their offspring may have of particular diseases. So I'm going to talk about a particular muscular dystrophy. We have to focus and cone down. Duchenne muscular dystrophy. Now Duchenne is a so-called X-linked disease. That means the mutation that causes the disease is located on the X chromosome. So Duchenne affects mainly young boys, whereas ladies are carriers of this particular illness. Okay. Sit on the front. Okay. Thank you. Are carriers of Duchenne muscular dystrophy. And the product of the gene that's involved in Duchenne muscular dystrophy is a protein called dystrophin, and it's a muscle. It's a protein that's expressed in muscle. If I turn those fibers, those blue fibers, so they're coming out of the screen towards you, you see a series of cross sections of cylinders, and this dark staining here, which is on the inside surface of those cylinders, is for the protein dystrophin. And you can see in Duchenne muscular dystrophy it's absent, and the tissue is significantly affected by that. We see a loss of fibers and a sort of fibrotic response in these affected tissue samples. There's also a lot of what's called inflammation. Cells of the immune system, these blue dots are shown here, infiltrating the tissue and causing more damage and more scarring. Now, the outcome of Duchenne muscular dystrophy is quite severe. It's a progressive muscle wasting disease. Starts about the age of 4 in a child that up to that stage looks relatively normal. Progressive loss of ability to walk and to move, affects lower and upper limbs, affects also the breathing muscles, so eventually patients with Duchenne muscular dystrophy need permanent respiratory support. And the body wall muscles that won't hold the spine straight result in something called scoliosis or distortion of the spine. And it's a life-threatening disease ultimately in the, well, nowadays in the early 20s and late 20s in these patients on respiratory support. So I wanted to touch briefly on 3 therapeutic strategies. I'm telling you about these because here at Royal Holloway we've contributed to them. Something called exon skipping, something called gene addition therapy, and something called genome surgery. Now, exon skipping, antisense therapy, and conceptually that's about trying to correct the genetic machine. We're not correcting the gene itself, we're affecting the genetic machinery. And we do this with short synthetic DNA sequences. These are called oligomers, and Raphael has referred to them before, but these sequences can identify and hybridize with genetic elements. For example, with something called an exon. As you know, exons are the coding parts of the gene, and they can actually block the use of a particular exon. So a so-called exon skipping approach to therapy. And if we take this approach in Duchenne muscular dystrophy, this is just an example of a clinical trial in that area where patients were either treated with this exon skipping oligonucleotide, and that's shown in this dark blue purple line, or there was a control group where treatment was delayed. And you can see with delay of treatment, the patient's ability to move gets worse and worse until the treatment begins to kick in. And this is a drug which is was originally called etiplursen, shown here, and is now actually a registered drug, certainly in the US, called Exondys 51. And there are others of this type coming through, again, as products of our research here at Royal Holloway. So exon skipping, let's look now at the gene addition concept. This is a little bit more obvious. We have a damaged gene. That gene is on the X chromosome. Can we perhaps substitute the damaged gene with a healthy version created in the laboratory? We can do genetic engineering nowadays, as many of you know. We can actually clone genes and amplify them in the context of bacterial carriers, such as E. coli, and this sort of plasmid type of DNA. So the question is, do we have lab-based healthy copies of the DMD gene? Well, yeah, we do. We've had them for many, many years, probably 15 or 20 years. They're called dystrophins, or so-called microdystrophin genes. So we have these genetic elements, we have the DNA. Can we deliver the DNA to patients? Well, actually, it's not as easy as you would think. When you're having lunch today, just think about it. You're eating DNA. Okay? If you have an apple, you've got apple DNA. So actually, our bodies are designed to resist DNA, genetic material, entering in a functional sense into our system. So we have to use a vector, a carrier. And that carrier really nowadays is several types of viruses, many of them harmless viruses, which are used to carry genetic material into tissues. And we use a virus, it's called AAV, adeno-associated virus, to deliver this microdystrophin gene into skeletal muscle. So does this process work to alleviate or prevent Duchenne muscular dystrophy? Well, first of all, we have to use animal models or veterinary disease models to test the therapy. That's obligatory these days, currently, and I'm not going to get into the debate about animal research or not animal research, but let me just say one thing. I don't think there's any section of society that values, honors animals more than the scientists that work on them and study biological phenomenon. So that's just a point to make. So do they work? Well, yes, they do, but so far proved in animal studies. In, for example, this little animal here, it's called an MDX mouse. It has the same genetic disease as patients with Duchenne muscular dystrophy. There's also a veterinary condition in golden retrievers, which again is identical. So it's a curative in the mouse and with very profound improvements in these veterinary studies. Now there are a number of clinical trials running now in the US, and very soon there'll be one running in the UK, of gene therapy for Duchenne muscular dystrophy. And currently, although these are really safety trials initially, there are some positive indications that gene therapy may be effective. Now I'm going to show some movies here. These are not scientifically, statistically relevant information. These are anecdotal observations of parents whose kids are involved in some of these trials. So this is a movie of a little boy with Duchenne muscular dystrophy, a certain age. This is before the gene therapy. You can see he's struggling to move upstairs. Here is the same young man after the gene therapy, and you can see anecdotally it looks like he's recovering a lot of muscle strength here. And then finally another example posted online of a patient treated in these gene therapy trials. So this looks pretty amazing to me actually, and so we're very hopeful that these positive indications will lead to a genuine authorized medicine, as Jane has spoken about, authorized gene therapy medicine for Duchenne muscular dystrophy. Now the final point I wanted to mention, because it's in the news a lot lately, is this idea of specific genome editing. We now have the tools. We have enzymes that are engineered and developed to identify and alter genetic material directly on the chromosome. So for example, in muscular dystrophy, this is just a schematic of the gene, but if you imagine there's a big section of the gene missing, that's the mutation between exon 44 and 53, then we can use these enzymes, these molecular scissors, to cleave the chromosome and encourage the introduction of an artificial genetic element encoding all of this missing genetic material. A corrected allele giving rise to the full-length protein being expressed, actually on the very position in the X chromosome that's affected in the condition. And again, here at Royal Holloway, we've been in the forefront of this. This is a couple of scientific publications describing that work, again, at this stage, in an experimental animal situation. Now this type of therapy is really, this type of genome correction therapy is really designed to target what I would call the stem cells in the body. And your muscle tissue actually does have stem cells as well as those long multinucleated muscle fibers that contract and produce muscle strength. If you damage your muscle, if you're in the gym too long, or you're training too much, or you have some other infectious damage to muscle, the muscle will be repaired by a stem cell population. And actually I just wanted to show you this in case any of you have not seen a stem cell before. So this is one of these muscle fibers, and up here and here we have some muscle stem cells that actually run around on the surface of the fiber. You can see them dividing 2 to 4 to 8 to so on, and these stem cells will repair any damage to those muscle fibers. So if we could genetically correct those stem cells, we have a mechanism, long-term mechanism of a genetic correction in patients with inherited diseases. So I'm finishing up now. This is actually, I think, very exciting times for gene-based medicines. There's been a revolution in our understanding of DNA, our ability to manipulate genes, but there's a new revolution coming in this century, a genomics revolution and a bioinformatics revolution. And hopefully some of you may be part of that revolution. I'd love to be here in 50 years' time to see actually what is being achieved in the genetics of disease therapy. And these are a few pictures of people from our labs, students and doctoral scientists working in the lab. This is actually a youngster with Duchenne muscular dystrophy that came in to see what it's like to work in a genetics research laboratory. And I'll finish there. Thank you. Thank you. Yes, of course, if you have time.
Yeah. So unusually we have finished early, so we have time for a few questions. So you are welcome to address any that you may have to either Jane or Professor Dixon or myself, if there is anything that is particularly important that you would like to address, this is the time. Anyone?
Yeah.
Just a quick question. You used the term bioinformatics in the previous slide. Can you just talk a little bit about what that means?
Yeah, bioinformatics. You know, in biology, or in biology in general, but maybe in medical research, there is this revolution I'm talking about coming down the line. One aspect of that, I think, is regenerative medicine, the ability to change tissues and regrow tissues and stimulate that or modify those tissues. The other revolution is this collection of genetic data. If we were sitting here in 10 years' time, maybe 15 years' time, probably the majority of people in the audience would have had their DNA sequenced, their genome, at least in part, sequenced. So that information will be available. It's a huge amount of information. Because while we talk about the human genome, actually, unless you have a twin, your genome is distinct from everyone else's genome. So there are billions of genomes out there, and that data needs to be collected in a mass. To analyze that data takes enormous computing power. What are the differences between individuals? How does it correlate with your known risk assessment for heart disease or diabetes? How does it correlate with your family history of conditions? Can it be used predictively? Do we want it to be used predictively? You know, in this day and age, all of the old guys at my age, we have our blood pressure and our cholesterol and maybe our glucose measured regularly. 20 years ago, a lot of people would have poo-pooed that. I don't want that stuff. Soon you will have your genome analysed and those risks assessed. So that level of analysis is enormous. It requires experts in computing, in algorithm development, and in data management and data storage, and it's just a massive industry developing in its own right.
So I touched on that as well a little bit when I mentioned artificial intelligence becoming something very important in medicine, the automated analysis of those data. So within the context of genomics research, the analysis of information is becoming more and more important. And many of us have wearables which register a number of health data that are being to our data providers. And again, that's continuous gathering of data that is analyzed and can become, it's already become part of routine healthcare. So the analysis of that data is more and more important, and it's massive in terms of the collection of data.
And as I was discussing with Jane before we gave the presentation, there are huge ethical issues associated with that. There are companies collecting genome data right now. There are companies like, dare I mention it, something like Ancestry.co.uk or 23andMe, who will take a sample of your saliva and then they will sequence your DNA and they will amass that data. But you begin to ask yourself the question, well, what would happen if a big insurance company like Aviva or others, I'm not mentioning them specifically, requested access to the 23andMe genome database? Well, you are applying for life insurance or a mortgage or whatever. So there are big ethical issues associated with that. I mentioned preconception screening. Would you, with your life partner, chosen life partner, like to know what are the possibilities of you as a couple passing on a genetic disease to your offspring? It doesn't affect you, it doesn't affect your partner, but together you could produce a recessive inherited condition. Would you like to know that or not? These are important questions that we really have to begin to start thinking about right now, because once genomes are sequenced, the information is available to you.
First, the little familiar with 100,000 genomes project
will offer whole genome sequencing to typically children, as well as children with cancers, and to people with certain categories of rare disease. So it's not as far away as you might think. You or someone in your family may be offered whole genome sequencing through the NHS. And one of the things that we hope is that when you have a genome sequence, you will contribute that data for social good to use in research. So just to point out that it's not a minister group, it's not science fiction, it is.
Yeah, we'll split the groups here. Thank you very much, everyone. Thank you to our speakers today for a very interesting session. And what we are going to do now is to tell you how we are going to split the cohort of visitors, so you go to either the lab activity or our exhibition, and then you will be swapping over. So the London Oratory School, St. John's School, and Heathland School will be our group A, and Yasmin here will take you to our labs to do the lab activity. And Trinity, West Thames, and any individual bookings will become Group B, and Sophia will take you to our exhibition. And in an hour time, we will swap over the groups. Thank you very much, everyone.